CBH. He serves as a tutor for the brain circuits cluster, helping others with their data analysis questions. He uses a combination of electrophysiology, computer vision, and artificial neural networks to explore how motor signals and behavior are encoded in the prefrontal cortex. And Adrian's going to give us a bit of a general introduction to DeepLabCut as well, so that the other speakers didn't have to spend too much time on that part. So Adrian's presentation will be about 15 minutes, and Ashley will come back into the picture after 15 minutes. So take it away, Adrian.
Great. All right, so just let me share my screen again. Let's see here, and this one here. All right. Okay, so I'm going to talk a little bit about automated behavioral classification, but the starting point will be DeepLabCut. Okay, so I guess we start with, what is DeepLabCut? Well, DeepLabCut is a means of getting away from the bad old days of manually annotating video frames. So here's just an example from some of our work of manually annotated videos tracking limbs in a rat in this case. Of course, this work is very painstaking and takes a very long time. And wouldn't it be nice if there was a way using all the sort of modern machine learning technology out there to automate this process? So along comes DeepLabCut. DeepLabCut is built on convolutional neural networks. I'm not going to go into too much detail about exactly how it works, although I'm happy to at another time. But so we use a very large pre-trained network, ResNet, which has been trained on all sorts of images, and then we specifically train a very small top layer for the specific tasks. So in the example here, this is tracking joint locations, rodent forelimb, but it can be trained for all sorts of things very, very simply. And this has been incredibly useful as a tool for researchers in terms of tracking features on animals for all sorts of experiments and all sorts of animals. And I encourage everyone who hasn't had a chance to have a look at the repository and all the sort of examples that this has been used on the repository from the Mathis lab, which I'm sure we'll have links to. Okay, so what does this look like in practice? Well, so here's some examples from some of the work from my lab. It just is a very simple case of playing in the background just of head tracking. So once the network's been trained, it's able to look at a video it hasn't seen before and generate coordinates, for, in this case, just for the nose of the animal as it runs around the enclosure. And in more complicated case here in the corner, it's tracking all the limbs indicated here, as well as nose and tail points. Right. So what do we get from DeepLabCut? Well, DeepLabCut outputs in what's .hdf, a hierarchical data format. So this is a matrix that contains both the coordinates of the features you've been tracking, as well as a likelihood number. So this likelihood is how certain the algorithm is of the predicted coordinates, or in other words, how well correlated the predicted location is with labels on similar inputs that it found in training. And if you look at these outputs, sometimes there are areas of significant artifacts where the algorithm is unsure. Perhaps the feature you were tracking is occluded or something like that. And so you may need to do a bit of post-processing on it, in this case, just a smoothed output to sort of solve these low likelihood areas. But there's all sorts of ways you can get around this. Okay, so what are some things you can do with this? You've got the limb location. So let's say you have some electrophysiology data from the same animal at the same time. In this case, this is recordings from the PFC. You can do some correlational analysis with neural data, which is what our lab did in this paper that we put out a couple of years ago, using some statistical techniques to sort of compare our neural signal with this automated limb location tracking. And you can do things like, we built a neural network which would predict limb location from neural signals, and then we could validate that with this automated annotation generated by DeepLabCut. But we can go beyond that. We can go beyond just pose and location information and start to think about behavior and complex behaviors. And again, we return to the sort of the old way of doing things manually, can be quite painstaking. And in behavior in particular, manual annotation is highly subjective and reliant on a priori definitions of what exactly constitutes behavior and what exactly constitutes the particular behaviors that you're interested in. So wouldn't it be nice if there was a way to automate this? Well, again, you know, there's several automated approaches that are gaining popularity these days. And these are broken up into supervised, unsupervised, and semi-supervised techniques. It's just based on the algorithms that underlie them. I'm going to talk briefly about a couple of them. So one automated semi-supervised behavioral segmentation approach called B-Soid, and then a bit more on an unsupervised behavioral annotation technique, Motion Mapper, and some supervised extensions to it. So B-Soid was developed to study naturalistic behaviors in open field experiments. And so it's a sort of multi-step process. It performs some initial dimensionality reduction and clustering using unsupervised techniques, and then it's piped to a downstream classifier. This is from DeepLabCut and is available on their GitHub. So I believe Ellen will be talking a little bit more in detail about a particular use case for B-Soid. So I'm not going to go into detail on it, but this is one approach that people are using. And another approach that people have been working with is something called Motion Mapper. And so this was originally developed to study behavior in fruit flies. There's paper there. And it sort of was built on this realization that behavior is multidimensional and temporally complex. And what do I mean by that? Well, what I mean is if you think about behaviors, let's go with our rodent example, which we've been carrying through this, a behavior like grooming. If you think about the features you're tracking, they all are moving at a different sort of stereotypical frequency given the behavior you're interested in. So in that example, the hind limbs might be moving very slowly or not at all. The forelimbs are moving quite rapidly as the animal grooms, and say a point like the head is moving more slowly. And so you've got this relationship of various features that you're tracking moving at different frequencies, and you can use that as a way to sort of identify a particular behavior. And that's done by something called phase-space analysis. So I'm going to give a slight warning here. This gets very mathy. I'm not going to go into the math right now in the interest of time and all of your sanity, but if anyone is interested in the details, I'm happy to talk about it more at another time. And so we sort of turn this dynamics question into a geometry problem via this phase-space analysis, via these mathematical techniques, because geometry problems are easier to think about, and we have many, many tools to solve them. So I'm just going to pop through exactly what's done, but what this is called is wavelet projection. And so we use a Fourier transform to project our signal into the complex plane, and we do some wavelet projections. And what we get for each feature that we're tracking is now, instead of just the location, we have this sort of expanded feature set, which is how that feature is moving in the various frequencies through time. And this is what we use to sort of divide up and categorize our behavior. So in Motion Mapper technique, we do some dimensionality reduction on this huge feature space using t-distributed stochastic neighborhood estimation. So it's just a graph-based dimensionality reduction. You could really use any method for this, although they will have, that will change your results. And if you graph the output of this, you get a sort of density graph of the feature space, and you can see you start to get some structure, and you can imagine how you could cluster this and sort of pull out distinct behaviors given this description. And so this can be done with something like the watershed transform. And so that labels, you get a labeled density graph like this of different behaviors. In our work, we've been using Gaussian mixture models instead. We found them to work a little better, but the technique is very, very similar. So, points are assigned to random clusters and then iteratively reassigned with gradient descent. And again, you get some labeled output of the behavioral space. And so the various points in time are divided into various behavioral clusters. And if you like, you can, then transform this into an ethogram or other behavioral display. If you want to sort of have a way to visualize behavior through time, sorts of things. It's all sorts of useful things that can be done. But in this last minute or so, I do want to mention that there are some significant drawbacks to this unsupervised approach to behavioral classification. So these clusters you get may not be relatable to human observable distinct behaviors. And what I mean by that is, if you looked at frames of video that were divided into two clusters automatically, you might not see as a human observer, well, why are these two separate clusters? Or what is this cluster, like what is all the behavior in this cluster? What behavior is that? What name would I give to it? So it's often very difficult to visualize this, and that makes it difficult to measure performance. And so some supervised validation is often required. And so in our case, this is just an example of something you could do. We replace the dimensionality reduction and clustering steps with an artificial neural network that we train via some manually annotated behavioral data using those DeepLabCut labels. And this makes it much easier to validate results and to visualize performance. And so you can, this is just a screenshot from a piece of software that I built for our data that allows us to select various models and play around with the parameters and see the frames of video that are put into each category. Or if you wanted, you could look at the activation maps of the neural network if that's of more interest. But so you can sort of build out these tools to enable you to visualize your output, and that allows makes validating this much easier. So hopefully that just gives you some ideas, an example of what can be done with DeepLabCut in a lab setting. And yeah, I hope that's piqued everyone's interest. And that's it for me for now.
Thank you so much, Adrian. Our next speaker is, and we're just moving on quickly for time. Our next speaker is DongSheng, and I believe Jeff will introduce him.
Hey everyone, just trying to find my notes again. So DongSheng trained as a doctor, where he did research and treatment in Parkinson's and other brain disorders using deep brain stimulation as a potential therapeutic. He's a postdoctoral fellow in the Murphy lab, and he's interested in the brain circuits that underlie sensory motor integration and motor functions. So he uses artificial intelligence and computer vision, including DeepLabCut, to do these things. So DongSheng, please start your presentation.
Hi, good afternoon, everyone. My name is DongSheng Xiao. Yeah, I will quickly go through three projects I have been using DeepLabCut in our lab. So the first project is real-time tracking. So we noticed this DeepLabCut will be a very useful tools. Just is posted on the archive. So it's before it's accepted by the Nature Neuroscience. So we quickly use these tools in our lab, and I work with IX students. He's the Brandon Freeze. He's the second year undergraduate students in UBC, and he's very talented. He helped us to develop this real-time tracking system, can track the body parts of the mouse with a very high speed and a short delay.
Excuse me, sorry to interrupt, but it seems like your slides are a bit cut off on the side. Do you mind expanding your field of view for us, please?
Oh, it's cut? Oh, sorry.
Yeah, that's okay, no problem. Perfect, thank you so much. We can see them all now.
You can see now? Okay.
Yeah, yeah.
Sorry. Okay, so we published this real-time tracking system on eNeuro, and I further developed this system, and I asked the question whether and how augmented sensory feedback can be designed to improve the learning of motor skills. So I trained the mouse to move his paw over a threshold to get a water reward. So after training, you can see the mouse indeed can increase his paw movements. But the problem is that the mouse only increase one. It will increase both paws movements after training. To solve this problem with the help of Pancache in our lab, we help to add the code the paw movements with auditory feedback. So when the mouse moves his paw, the tracking signal will trigger a different frequency of sound. And then we train the mouse to do the same task. You can see. Oh, I don't know why I cannot play the videos. But after training, the mouse can quickly learn this task and can only move his one paw to get a water reward. And you can see the performance is quite good. Sorry, cannot play the video now. I don't know. I just don't know why it cannot play. Cannot play it. Oh, that's bad. We are further develop this system to add more features. And another projects in our lab is a 3D word mouse. We use create a 3D word mouse to generate synthetic training data for behavior analysis. The idea of this 3D word mouse comes from a game project. So when generating synthetic realistic behavior videos from brain activities using a game model. So when we compare the fake video and the real video, we think about to transfer the behavior video to a 3D virtual mouse maybe, and then to better classify the behavior, because the different mouse in the behavior video will have different shape and size and different camera angles. If we have this 3D virtual mouse, will be remove these barriers. So another very talented undergraduate students, Louis, help us to build this very cool 3D virtual mouse. And this really help us to get a better synthetic training data, train the DeepLabCut model to get a better performance. You can see, seems all the video cannot play, but we can easily get a 3D 3D pose from 2D cameras. So using this 3D virtual mouse. I think Xiao will present more on these projects. So so we also can use this 3D tracks to better classify the behavior into different behavioral state. Another project I use DeepLabCut is MesoNet. So DeepLabCut is very general deep learning model. So we also can apply this model to other data sets instead of behavior videos. We can use on the calcium images, for example. So I train the DeepLabCut on a lot of brain images to predict landmarks on the brain. So you can see after training, we can get a very good performance to predict the landmarks compared to the human labelers. And combined with another machine learning model, UNet, we can automatically rescue the atlas to the brain and then segment the brain to different brain regions. You can see when we overlay the brain atlas to the image to the sensory mapping data, the brain regions is aligned well with the sensory maps. So in addition to rescue the atlas to brain, we also can rescue the brain to a common atlas. So after transform the brain, we can better align the brain image in a same common coordinate system, and then to better classify the brain data, quantify the brain data. For example, this week transform the brain image to a common atlas, and then to classify the cortical activity motifs. We can get a better motif pattern after this transformation. So sorry about the videos. Yeah, that's all. Thanks.
Thank you so much, DongSheng. It's all right. Sometimes there's technical issues. No worries at all. I think our next presenter is Ellen, and Jeff is going to introduce her.
So Ellen is our next speaker. She is a PhD student in the Raymond Lab at the DMCBH, and also a Canadian Open Neuroscience Platform Scholar. Her project is studying how corticostriatal signaling is altered in Huntington's disease and how these neuropathological changes contribute to motor behavior and motor learning deficits. She's exploring this using a combination of in vitro and in vivo methods, including fiber photometry and, of course, some DeepLabCut. So, Ellen, please go ahead and share your screen and start your presentation.
Great. Thank you, Jeff, for the introduction. Okay, so you can see my screen, right? My presentation. Good. Okay. All right, so today I'm just going to talk about, give you a snapshot of a few different ways that I'm using DeepLabCut to analyze the data in my project. And just a very quick background on my project. So in the Raymond Lab, we study Huntington's disease, which is a neurodegenerative movement disorder. And the striatum is the main site of, well, the primary site of degeneration in this disease. And that's a brain area highly involved in movement and in motor learning. I'm using fiber photometry, which is a technique that enables in vivo imaging of deep brain structures like the striatum during freely moving behavior. And I'm using DeepLabCut along with other methods to examine motor behavior and kinematics in the wild type mice as well as HD mouse models, and combining this with fiber photometry to explore how GCAMP, the calcium sensor activity in the striatum, correlates with motor learning and spontaneous behavior. So one of the behaviors that I've looked at is the open field. So this is an example of a mouse in the open field. And over here in the corner, this is how these are the coordinates that I trained DeepLabCut on. So I labeled all of these different body parts. And then you can see this video on the right showing the movement of the mouse in the open field. And then I'm also showing you here the fiber photometry GCAMP data at the same time during this behavior. And so here's a few just different examples of the different kind of behavior patterns you can see from mice in the open field. And using this, these coordinates, I've taken the DeepLabCut coordinates and then from there, analyze the coordinates to extract different measures such as total distance traveled, time in the center versus the edges, time in different quadrants of the open field. And just as an example here, this is showing the wild type data and the Huntington model called YAC128 at an early stage in the disease. And overall, I saw a slight increase in the total distance traveled in the Huntington model. And then looking at the total distance traveled per minute, so in a 10-minute period, how much they traveled per minute, you can see that over time in the open field, they decrease their total distance traveled as they get habituated. And if you compare the first day they're in the open field to the fourth day, there's a reduction in their distance traveled. So these are some measures that you can easily get with DeepLabCut and some analysis post DeepLabCut. And then I also have correlated these measures with the GCAMP recordings in the striatum. So this is an example of a wild type recording and a Huntington mouse recording. And I'm using MATLAB and using the peak finder in MATLAB to identify all of the peaks in the GCamp recording. And what I'm trying to show here is that in this example from a Huntington mouse, there's actually a higher peak frequency while they're exploring the open field compared to the wild type example. And one thing I've done with a lot of my different behavioral measures is I've looked at the correlation between the peak frequency and the behavior. So in this case, total distance traveled. And I found that in wild type, in this case, the total distance traveled, as the mice are more active in the open field, there's increased peak frequency, so more activity in the striatum. Whereas in the YAC128 Huntington mice in this example, there's actually no correlation between the two measures. So this is one way in which it seems that the activity in the striatum is kind of not corresponding the same way to the behavior as it is in wild type. And I've also done some more sophisticated analysis with DeepLabCut and B-Soid that Adrian mentioned. And so this is a behavior classification software that you can feed your DeepLabCut coordinates into and then look at, basically it will identify different behaviors based on the coordinates that you give it. So based on the position, the angle, and the speed, and different characteristics of the labels. And I'm not going to go into detail just for time, but if you're interested, you can ask me later about some of the preliminary data that I've gotten from this analysis. But you can look at things like the types and frequencies of behaviors, behavior transition probabilities, and kinematics of body parts during behaviors using this analysis. And this is just an example of some of the newer videos I'm taking now from below, which is a great way to be able to look at the pause and look at the kinematics of the pause. Okay, so then for the rest of the presentation, I'm going to talk about my analysis of the accelerating rotor rod. So this is a task where the mice learn to stay on a rod that's rotating, and it increases in the speed of the rotation, and the mouse has to try to stay on the rod. And then the latency to fall is measured. And as they learn this task over 4 days of training, there's an increase in the latency to fall. And so what I noticed from my preliminary analysis is that early stage YAC128 Huntington's mice didn't show any difference in latency to fall. But I was interested in looking at the kinematics of the pause and seeing how that changes over learning. So to do this, I use DeepLabCut. And here I'm showing you an example of a mouse showing the Y coordinates of both the right and left foot on the rotor rod. And then the black lines are showing where the rotor rod, the top and the bottom of the rod. And then this is over time. So you can see that as the rotor rod accelerates over time, the steps of the mice get larger over time, and they even start to slip below the rotor rod as they near falling off. And then this at the end is when the mouse fell off the rotor rod. And here are just a few examples of variability in the data. So you can see this mouse, even though it stayed on for a while, it slipped a lot, whereas other mice only slip right before they fall off. So one way I've analyzed this is by looking at the foot slips. So using the peak finder in MATLAB, I've used this to identify the foot slips that happen in time. So to do this, I flip my data over so that it's upside down here, and 0 corresponds to the bottom of the rotor rod. And then I use a threshold of 0 and identify any peaks that are above 0, meaning that these are when the foot is below the rotor rod. And then I count how many times the mouse slipped. And interestingly, I found that although these mice are performing equally well as far as latency to fall goes, they have an increased number of foot slips per minute on both day 3 and 4, and especially on day 3. So the Huntington's mice, even though they seem to be doing just as well at the task, they're actually slipping a lot more and have this motor coordination deficit. And another way to look at this is looking at the cumulative foot slips over time. So you can see that after about 150 seconds here, the wild type is slowly increasing the number of foot slips as the rotarod accelerates, but there's this huge increase in the Huntington model. And then one other thing that I also looked at with this analysis is looking at the height of the pause over time on the rotarod. And you can see by comparing day 1 to day 4 that by day 4, the average height on the rotarod has increased, and the data is also a lot tighter. The variability is also increased as they've learned to do this task. So I decided to look at the successful trials on day 4 when the mice stay on the task for the entire duration of time. And I found that the height of comparing wild type to Huntington's disease, the height of the pause in the rotarod was similar for the right and left paw, but there is an increase in the variability, so the standard deviation in the Huntington's model. So showing another kind of subtle difference between these mice, even though they're still staying on the rod for the same amount of time. And then the last thing I'll mention, which I'm working on right now, is now bringing the GCamp data back in and looking at the activity in the striatum. And one thing I'm working on is lining up the GCamp signal to the foot slips. So this is an example from one mouse. The red line is when a foot slip happens. And then I've lined up every single time the mouse has a foot slip, I've lined up the GCamp data to the red line. And you'll notice there's a lot of variability, but I noticed that overall there seems to be an increase in activity following the foot slip, whereas when you just line up to random time points, you don't see this, this same pattern. So this is an interesting analysis that I'm currently working on. So in summary, DeepLabCut tracking and programs such as B-Soid are a way that allow for detailed analysis of behavioral patterns and limb kinematics during behaviors in the open field. And these softwares also save you a lot of time. Using tracking coordinates from DeepLabCut, I was able to find significant changes in paw kinematics during the accelerating rotarod in HD mice. And all of these analyses can be combined with photometry or with other imaging or recording methods so that you can correlate the neural activity in the brain to behavior and look at how this is affected in disease models. So thank you for listening.
Thank you so much. I believe our next speaker is Xiao, and Jeff will introduce him now.
So Xiao is a PhD student in the Murphy lab at the DMCBH. His project is focusing on the quantification and correlation of behavior and brain activity. And he's using recent machine learning tools to do this, including DeepLabCut and what he's about to tell you about. So Xiao, please share your screen and begin.
Okay. Can you see my screen now? Or
Yep, we can see it.
okay, great. Thank you. Thanks, Jeff, for introduction. Good afternoon, everyone. Today my topic is data augmentation using the synthetic animated mouse. And I will focus on the 2D case because it's more related to our general topic today. During the past half an hour, Adrian, DongSheng, and Alan already gave great presentations that contain the information about the DeepLabCut in details, like what the DeepLabCut is, what kinds of application can be done, and how powerful the DeepLabCut is. In short, the DeepLabCut is an accurate and flexible toolbox. It can identify various body parts in multiple species and multiple behavior, if you provide it enough training data. However, there's no free lunch. A toolbox can fit every situation always means it cannot fit any situation out of a box. A well-trained DeepLabCut model typically requires 50 to 200 human-labeled frames. If we assume that there are certain points need to be labeled for each frame, like this figure, the mouse left paw, it will be 2,600 points need to be labeled in total. Even experienced DeepLabCut labeler will spend about 4 hours for labeling it. So the data and label are very costly. But like many other groups, our labs spend the most of time dealing with the mice model and its experiment data. Therefore, we're willing to trade the flexibility across species for reducing the user effort to label the training images. Is there any way to get the data and label without any user effort if the species is fixed? The answer is yes. A recent technique called simulation and learning catches our eye. Instead of gathering the data and labeling them manually, they simulate lots of automatic labeled synthetic data using computer game like this one. Because it's a computer game, the software will tell you the label for each object and where is the boundary of each object. They can also specify the scenario to compensate missing edge cases. Like this one, the simulation makes it possible to generate many dangerous scenarios like crashing, which is hardly be seen in normal life. And however, there's no existing video game to generate the behavior of mouse. That's why we built a synthetic animated mouse based on CT scan images in an open source 3D creation suite called Blender. Our synthetic animated mouse is an open source, high resolution, realistic model of the mouse skeleton, skin, whisker, and fur. So I will show you the video. This video clearly shows, this video shows how the synthetic animated mouse model works. And if you want to generate the movement, we only need to click on the body parts you want to control and drag it to the position you want. During the simulation, we can also introduce additional diversity by varying the camera position, camera angle, lighting, and the surrounding experiment devices. So now we've got lots of automatic label data. If we want to use them for training DeepLabCut, the next step will be make it more realistic. We adapt the leading techniques called image domain transformation. This method enables machine to have human-like imagination abilities. For example, it can be used to increase the resolution of image, which is called super resolution, or make a photo like looks like a painting, or maybe change the weather from sunny to rainy. More surprisingly, it can transfer one's facial expression to other people. In this project, we are using this method, image domain transformation algorithm called You Got It, to transfer the style, such as the texture, lighting, and background from the real mouse video onto our synthetic mouse video. So this figure illustrates the step-by-step procedure for data augmentation using the synthetic animated mouse. Firstly, we generate lots of automatically labeled images by simulation here, and because it's simulation, so the label can be exported as a CSV file, which has exactly same format as manual label frame in DeepLabCut. In this situation, the labels refer to coordinates of each body parts. Then we can use the You Got It model to transfer our simulated animation into more realistic video, and combine. And then we can pack those images with labels and send them to DeepLabCut to train a DeepLabCut model. After the model is well trained, we can apply the model to the real unlabeled data and get the coordinates of our desired body parts. Here are some videos shows the performance of our image transformation algorithm. And here are some more videos shows the performance of the image domain transformation algorithm. If you compare the Ugalit style transfer video with the real reference, I think the difference between them are relatively small. Yeah, and we also use some statistic method to measure the difference between two groups of images. One is the synthetic animated image set. The other is a real reference. If we look at the blue box at the left panel, the SSIM, which is a structure similarity index measure, histogram shows that the difference between synthetic animated image after the image domain transfer and the real reference image is similar to the difference between two real reference images. So we also compare the performance of DeepLabCut, which is trained with manually labeled frame, and the model which trained with our synthetic animated frames, as shown in the yellow box here. The DeepLabCut trained with synthetic data reaches a very similar performance as a model trained with 200 manually labeled frames. So we think it will, it can significantly reduce the human labor. And then after that, to show the potential of our synthetic mouse model, we to show the potential of our synthetic model, like how to compensate missing edge cases or further automation, I applied the data of movement from a huge famous human movement library, which called mocap, to our mouse model. We successfully generated some video like the walking and jumping. We believe that this synthetic model can benefit more study for following reasons. Firstly, it can be controlled with any existing data like mocap. Secondly, it can work seamlessly along with any Python script or machine learning algorithm which written by PyTorch. Yeah, finished. Thank you.
Thank you so much, Xiao. Now we'll have a short questions period. Does anyone have any questions? Oh, go ahead.
Yeah, I was just going to invite everyone to ask their questions. You can type them in the chat or just raise your hand with the raise hand function in the participant menu, or unmute and ask. While people are thinking of questions, Tim, did you want to say anything summarizing all of this?
Well, I think first just to thank all four speakers, and just to let people know that I mean we're here at UBC, and you know, it's not limited to just today. We can, you know, this is to start further collaborations. And I think this is really an exciting tool. There's also a whole raft of information on the Mathis website, which is really useful, that I would also direct you to, and YouTube videos and neuron primers and things. So I'll just I'll leave it up to the trainees to ask some questions. Thanks, Jeff.
Sure. So there's a question in the chat for Ellen. Sarah, did you want to unmute and read out your question, or do you want me to do it?
No, I don't mind. No problem at all. So Ellen, I was wondering, you were showing some behavior from your rotor-rod in your open field test. How long did it take to train the software for that? And do you have any additional behavior tests that you use in your day-to-day that you don't mention in the presentation?
Yeah, that's a good question. So I'm trying to remember. I think it did take me a few iterations to train DeepLabCut. So I train it and then I see how well it did. And I wanted to get it, it's already pretty good the first time, but I'm usually training it a lot to get it really, really good. So I think it took me, I think I did end up training it on at least 300 frames. So I think about 3 iterations that I did. So 3 training sessions where I refined my labels or added new labels in and trained it again. Right now, I'm focusing mostly on rotor-rod and open field for the behavioral tests that I'm doing. I'm also doing a little bit of water T-maze. So I might end up using DeepLabCut as well. It's actually easy to just manually watch the videos for that one for the analysis we're doing. So we haven't used DeepLabCut yet, but we might also use DeepLabCut for a water T-maze where the mouse has to learn to turn a certain direction to find a platform.
If okay with you, I might just email you to the side. There's a lot of memory, cognition, anxiety, locomotor tests that we're doing as well, and other things that I'll be talking about later on. I think it could be kind of interesting to go back and forth and see what you're doing, what I'm doing, and how that could relate to some of the software that we're using compared to DeepLabCut.
Yeah, sounds great. Thanks,
Cool.
Sarah.
Anyone else have questions? So there's a question that popped up in the chat. Did you want to unmute and say your question, or do you want me to read it out?
You can read it out. Thank you.
Okay. So the question is, can we use DeepLabCut for multiple humans and animals at the same time? And can we extract distances between them? Anyone want to tackle that? Multiple animals, multiple humans.
Sure. Yeah, I can hop in. So the short answer to that is yes, certainly. So there's a couple of this being done with multiple mice in open field, as well as with other species. So this is certainly doable. One thing that may throw a wrench into that is if you need to keep track of which individual is which. So that may require some additional apparatus or some additional post-processing. So I think a number of people who are doing this, and I know that some people are doing these sorts of things at UBC use an infrared tracker or use an RFID tag to identify the particular animals, and then once you know which animal is which, then tracking multiple animals at the same time is no problem at all for DeepLabCut.
Thank you.
How are we doing on time, Ashley?
I think we're right on the ball here, about 5 minutes off of the 1 hour, about 5 minutes off the 1 hour mark. So I think we can, unless there's any other questions, I think we can move forward.
Okay.
Yeah.
So part 2 of this is an attempt to do a virtual hands-on DeepLabCut demonstration / guided tutorial. So we have a core facility in the DMCBH where we have a bunch of computers with GPUs. That's where I am today. And so Ashley and I have set each of these up with DeepLabCut and tested it on the data that was provided by Sarah from ICORD. And the idea is we will break into breakout rooms and one person will join, remotely access these computers, and we'll have a small group to go through the guided tutorial together. Ashley and I and others will jump between the breakout rooms to try and help you guys if you get stuck. And after Sarah introduces the data that she provided, Ashley will show you guys the first few steps just to get you going before we ask Kate and Megan to set up the breakout rooms. So with that, Sarah, maybe you can talk a little bit about the data that you provided.
Yeah, for sure. I have a
tutorial.
Absolutely. I have a quick screen that I'm going to bring up just to do a basic explanation of what the test is. So one moment while I get that ready. Okay. And there we go. All right. So the test that I'm looking into is essentially looking at memory in mice. So the data that we'll be looking at today is mice that are aged. So they're 15 months old at the time that they got a spinal cord injury. And this is 3 months later. So these mice are technically 18 months old, which anyone that works with rodents know that that's not a super common age that we usually work with. But essentially the concept is these animals are going to be put into a chamber and they're going to be given 2 objects of equal size, similar to the mouse essentially, and they'll be allowed to explore that for 10 minutes. And this is their kind of habituation. After that, they're going to be taken out for 10 minutes and let to rest in their home cage just to get a little breather and to kind of let that memory sink in of what they were just seeing. We'll be placed back into their cage where they're going to have to face the object location task. So essentially one of the objects is going to be moved and they're going to go around. As a concept, these animals should be more interested in the object that moved if they can maintain the memory. Something has changed and they should intrinsically want to explore that. After that, again, they're going to be taken out, another 10 minutes, placed back into their home cage, or eventually they'll be placed back in one final time, and the object that had not been moved previously will now be replaced. So again, we have something new that the mouse should intrinsically want to explore. So normally this would be done manually, which again, most people that have worked with animal behaviors would know that that's quite a pain to be able to assess where the animal's looking at, if they're in the proper orientation, if they're close enough, which is one of the things that DeepLabCut is going to be great in general to help cut down on. So that's the basics.
Okay, thanks, Sarah.
Yeah, awesome.
So,
and now, oh.
Yeah, I was just going to say, go ahead.
Oh, sure. Sorry, it's hard with Zoom sometimes. I'm going to give you guys a brief introduction in the first couple of steps of our tutorial. So I'm going to share my screen now. So once you get into your breakout rooms, somebody within your breakout rooms is going to download this program called AnyDesk, which I'm currently using, and then it will give you a number for each of the computers, and then kind of like we're doing here, one person will be the driver, so to speak, and then the rest of you guys will watch and give some feedback. So I'm going to walk you through the first couple of steps. So here is part 2.1. There's actually goes over steps 1 through 6 quickly, the breakout room list. So step 1 is to go to this OSF page. The link is provided for you. I already have it up over here. So then I'm going to scroll down. This is the data that Sarah Wheeler has kindly provided us. I'm going to take this folder. I'm going to where is it? Oh, there we go. Download as zip. So once that's downloading, okay. So it's going to download into a zip file. Going to be in our downloads. Here we go, crop video zip. And we're going to extract to, I'll extract here. Sounds good. So now, oh, did it name something different? That's okay. Once you get into your crop videos, oh, wrong folder. Here we have the 4 videos that Sarah so kindly provided us. So now I'm going to go reference back to my sheet here Scroll down a little bit. Crop videos. Next, I'm going to rename this for naming purposes. So I have a feeling this is going to throw some issues. So now we're going to do some terminal work. So if you haven't worked with a terminal before, if you haven't worked with a terminal in Linux, no worries. There's going to be lots of people to be able to help you out here. So now I'm going to open up our terminal and we're going to type in the words conda activate DLC GPU. DLC GPU. Now we're in the DLC GPU environment. Now we're going to enter the IPython environment. It's going to look like this. Now you're going to type in import DeepLabCut. Okay. So the next part is you're going to go back to files and you're going to go to home and you're going to go to DLC working directory. And you're going to select, you're going to either make a new folder because some of them might already have folders in there, or you're going to find another place that you want to put it. So I'm going to put it here because it seems pretty clean and nice. So when I go to step 6, this is where the magic happens. And this is where the start of your project is going to be created. So you're going to type in, just like what you see here, type in DeepLabCut dot create _new _project. Open parentheses, you're going to put the name of yourself, so, or whatever the name of the project, sorry. So I'm going to put it test _workshop. You want to close hypotheses, not hypotheses. I can't remember the name right now. And then I'll put my name. And then you're going to put the directory of the files. So this is the directory of where we extracted the video files. Usually you can do multiple videos for different training sets, but for the interest of this workshop, in the interest of making everything go a little bit faster, we're going to have it just be, we're just going to do one. I would suggest videos 602 or 604, because those have the clearest mice and easier for labeling. So you can click right and go into the terminal, click pwd. This is the directory that you're going to use in your other terminal. Sorry. Oh, another chat. Oh, thank you. Sometimes another issue with coding is sometimes when you spell things wrong. Oh, my AnyDesk is frozen. Oh dear. Let me just give it a sec. It's frozen on me. Okay. All right, it seems to be up and working again. Sorry, sometimes AnyDesk freezes. Okay. Now I'm gonna type in the file path, B0 lab, downloads, cropped video, spelled video wrong, 00604.avi, close bracket, BCR lab downloads. And now we're going to set the working directory, which is where we chose before, working directory equal to home, BCR lab, DLC _working _dir think, and then the last little bit, look down here, you want to have comma, copy underscore videos equals true. So hopefully I've spelt everything right.
There's a mistake, Ashley, in working directory.
Okay. What did I spell
Just
wrong?
directory wrong.
Oh,
DIRE, you must need.
oh, thank you.
Yeah, you'll just get an error.
Yeah, you'll just get an error and it'll tell you that it can't find it or something, but
Also, a slash missing between downloads and cropped.
thank you. The angle I'm looking at. The wonders of,
Yeah.
there we go. Thank you guys. Awesome. So this is about how, as far as I want to walk you guys up through on here. Hopefully, if you're not super familiar with coding or working with a terminal,